Good day, everyone. We're Group 12 of LBYKM41 NO1B, and we're here to present our protein of interest in our report entitled The Toxic Paradox, Botulinum Neurotoxin. Known as one of the deadliest toxin on Earth, botulinum neurotoxin is a toxin which originates from the gram-positive bacteria Clostridium botulinum, known for causing muscular paralysis. With this, it can be said to be a weapon of mass destruction when placed into the wrong hands, as its airborne release may lead to respiratory paralysis. More toxic than cyanide? Yes. Botulinum neurotoxin has an LD value of 1 nanogram per kilogram, meaning just a tiny amount of it, roughly the size of a grain of sand, has the potential to kill a whopping 9,600 people. So how prevalent is the bacteria causing it, and what are the types of BOT-N out there? Well, the bacteria can be found in most aquatic and terrestrial environments, and BOT-N is actually a protein, despite its menacing name. It has 7 types, 8 if you would consider the 2 subtypes of type C, and are known to be serologically and antigenically different, yet structurally similar. Types A, B, E, and rarely F are said to cause botulism in humans, while the other types in other organisms. Given the deadly nature of this protein, we humans have found a great way to deal with it. In fact, so great that we inject it to ourselves. Due to very small amounts and careful processing, the medical and cosmetic field actually make use of BOT-N type A and takes advantage of the limping effect it can do to the muscles. Aside from cosmetic applications, hence Botox, botulinum neurotoxin A can be used for other muscular problems and even chronic diseases. With this, let's now dive deeper into the very interesting protein that is botulinum neurotoxin type A.
Botulinum toxin is produced by the bacterium Clostridium botulinum as a single chain composed of 100 kilodaltons of a heavy chain and 50 kilodaltons of a light chain. These two parts of the single chain are linked by a disulfide bond, which, just like insulin among other proteins, get cleaved before turning into their mature state and performing their intended function with less amino acids as compared to their original state. It has been found that the protein's function can be classified in multiple ways. It acts as a hydrolase, which means it destroys bonds by using water in its catalytic activity. It acts as an endopeptidase, catalyzing peptide cleavage within the chain, which is to say it cleaves non-terminally. Lastly, it is considered a metalloproteinase because it involves a metal in its catalysis. Here are its other mesh classifications. Maximum likelihood and neighbor joining phylogenetic analyses yielded these results. ORF1 and ORF2 were found to be related to BONT and NTNH, but they diverged before expanding in Clostridium. These two are then multi-domain homologs of Clostridial neurotoxin, as shown by the right figure. Furthermore, due to the nature of the divergence, it is likely that they originated from lateral transfer from a species distinct from Clostridium botulinum. In addition to this, the amino acid sequence of botulinum toxin A has been found to have a great homology with that of tetanus neurotoxin, shown at the lower left part of the screen, which causes the fatal disease, tetanus. As for the location of the chromosome, this was the gene itself that we found, but it is not numbered since bacteria don't have chromosomes. These numbers here indicate exactly where in the gene the sequence is found. This is the amino acid sequence for the protein and can be found using its protein ID, while this is the DNA sequence of the protein. This can be found by using known segments of the protein. Take note that using segments will yield multiple results coming from similar proteins, and that the longer the known segment is, the more probable it is that you are going to find the right protein. Now, here is the actual 3D structure of the protein. Let us zoom in and study its characteristics. Somehow, there is a regularity in the red area when it comes to every third or fourth amino acid, in that these have the same polarity. Furthermore, proline and glycine, which are characteristic amino acids for tight turns, are nowhere to be found. These properties are characteristic of alpha helices. Now we can use data banks to further explore the protein. For example, if we click on a particular part at the left, it would show us exactly where these amino acids reside in the protein.
So we now focus on the structure of the protein. From the protein data bank, we can search for botulinum neurotoxin A using its accession code 3BTA. Its structure has been determined using X-ray diffraction. And as we can see, the biological assembly, which is the functional form of the molecule, is the same as its asymmetric unit. Zooming in, we can see that the protein is subdivided into three domains. The receptor binding domain has two distinct subdomains that are comparable in size. The C-terminal shown in red and the N-terminal shown in orange. Both are predominantly composed of beta sheets, the N-terminal subdomain being in a jelly roll motif and the C-terminal adopting a modified beta trefoil fold with a six-stranded beta barrel and a beta hair pin triplet. The most noticeable structural feature of the translocation domain, shown in yellow, is the presence of a pair of long antiparallel alpha helices that twist around each other like a coiled coil. This structure has been shown to increase the permeability of lipid bilayers. Both the translocation and binding domains make up the heavy chain, whose function will be discussed later on. Lastly, the catalytic domain in blue, which represents the light chain. It is a mixture of alpha and beta helices. Its notable feature is its active site, in which it has a catalytic zinc embedded within, responsible for the cleavage of an important protein in muscle contraction, which subsequently may cause paralysis.
But how does this enzyme cause death? You see, muscles are vital to life. Even heart is a muscle. And the neurotransmitter that's responsible for muscle contraction is acetylcholine. The vesicle containing acetylcholine binds to three specific proteins known as the snare proteins, which are all essential for synaptic vesicle fusion. The acetylcholine gets released and then binds to receptors on the other side of the junction, which through a number of reactions causes muscle contraction. Botulinum neurotoxin A is again composed of a light chain and a heavy chain. The binding domain attaches itself to the presynaptic nerve ending, and the translocation domain facilitates endocytosis. Now that the toxin is enclosed in a vesicle, the heavy chain assists in pore formation for the light chain to be released into the cytosol. The light chain, which contains the active site of the protein, cleaves one of the snare proteins, the SNAP25, which then hinders the release of the neurotransmitter out of the junction. This will inhibit muscle contraction. Through this mechanism, small amounts injected to specific parts of the facial muscles can reduce frown lines and wrinkles. But through the same mechanism, paralysis and subsequent stopping of the heart awaits.
What's next? With our discussion done, botulinum neurotoxin is just one of the plethora of proteins out there. And from our analysis, it just shows how complex these macromolecules are. From this pandemic to Botox, birth to evolution, proteins are one of the building blocks of life, and our ingenuity as humans to make the most out of it gives us all the excitement of where we can push the boundaries of science next. That's all for today, and once again, we're Group 12, and wishing you a good day ahead. Thank you.